Hey guys, just wanted to run through the notes with you. I can't believe this is our last unit, but here it goes. So, genetics is going to be the branch of biology that deals with heredity and the variation of organisms. What that means is the traits that make you you, and how those get passed down from parent to offspring. That's what we're going to be looking at. We started with mitosis, which was cells making new cells, and then meiosis, which was how we make those sex cells that carry those traits, how they're passed on. Now we're going to be looking at how those traits are then kind of passed even further. And so chromosomes are going to be what carry that hereditary information in the form of genes. And once again, that's determined by meiosis. It's going to be what's responsible for making you, for shaping you, things like that. Every living thing is going to have a set of characteristics that they are going to inherit from their parent or parents. Parent would be asexual, parents would be sexual reproduction. The way that we look at this is through genetics. It's just kind of understanding, like I said, how those traits are passed through offspring. The guy that's the big guy in science here, Gregor Mendel, he's going to be our father of genetics, or the father of modern genetics. He was an Austrian monk. And after he became a priest, while he was there at the monastery, he began teaching classes and studying math and science. He's also going to be over the garden, where he's going to get to work with some pea plants. And then he's going to get to use the scientific method and test out this theory on how traits are passed through parent to offspring. And so while he was doing the method, he went to reproduce his results, because that's a very vital step in terms of making a theory or coming up with a conclusion, is that your results have to be repeatable and reproducible. And he couldn't do that because he used a different type of plant. He went from using sexual plant to asexual, and then it just messed his results up. And so they kind of get vastly forgotten for about 100 years until someone else figures out what he did wrong and then goes back and gives him the credit. But he's going to determine how genes or traits are passed using cross-pollination with pea plants. And that allowed him to come up with three principles. The principle of dominance, which says one allele is going to be dominant or mask the expression or presence of another trait, which will be the recessive trait. And so you'll always have one trait that's dominant over the other, unless they're both recessive, and then the recessive gene will show. But anytime that there is a dominant and a recessive, the dominant is always going to show over the recessive. In the principle of segregation, this is going to be when the gametes are formed, the genes are going to become separated so that the sex cells receive each of one kind of gene. Remember, you have the homologous chromosomes that are the same. As they split to go to the new cells, that's just making sure that you don't have a cell that's missing a chromosome of any kind. And then the last one is principle of independent assortment, which goes back to that metaphase one. Anaphase one deal where we're independently splitting these up, lining them up, separating them. The members are going to segregate independently from other gene pairs during gamete formation. And while it has the term segregate in it, it is not segregation. Make sure you look for that word independently, so that it's principle of independent assortment when you're looking out for that on a test. Genetic crossing, this is going to be the bulk of what we're going to be talking about. Most of this is just kind of definitions type stuff. Our original organisms, the two that we're beginning to cross, they're going to be the P1 or the parental generation. And then what comes next is the F1 or first filial generation. That's the offspring of the P generation. Filial there meaning Latin for son or daughter. That's where that word comes from. And then in animals and plants, hopefully not humans, within that same group of offspring, if we were to take those offspring and make them reproduce, that would be the F2 or the second filial generation. Genes are going to be the factors that are passed from the parent to the offspring. And then the genes will be represented by alleles. They're the different forms of the genes, whether it's dominant or recessive. You can either have a homozygous dominant, where they're both going to be capital letters, homozygous recessive, or they'll both be little letters. Homozygous meaning the same. So whatever version of it, dominant or recessive, that you have the two of the same Heterozygous means different, and so you'll always have a dominant and a recessive, a big and a little letter. When we talk about genotypes, we're going to be referring to the genetic makeup or the letters themselves, the actual code that tells you what's going to be the physical appearance. The phenotype is the actual physical condition, so what we can see, the observable trait that is determined by the genotype. And then the way you could use this method here. It gets real confusing, so we're going to use Punnett squares. Reginald Punnett is the guy that came up with this tool to help cross traits, which is then used to help determine the probability of genetic outcome. So what are the chances that, if we have two kinds of genes, what is the offspring going to look like? And so this, when we do a cross, it's for one offspring. It's not saying that you're going to have 4 offspring every time. That's not what that's saying, but there's a probability or a chance. Because when Mendel was doing his study, he found that there was a 75-25 physical expression here. And so we get a similar outcome each time with the Punnett square. So you're going to get some kind of problem where it's going to break down, tell you that it says, write the genotypes of the 2 organisms that will serve as parents in a cross. But this one, we're looking at male and female osprey, which are fish hawks, and we're looking for large beaks. If they're both going to have large beaks, that means I'm assuming that the big B here is going to be for large beak. The little b will be for a small beak. So we have the 2 parents. We separate out the alleles. We can then place those alleles each by a box of the Punnett square. When we do that, we're then going to drop and bring our letters over to fill in for the box. You can see how I moved those there. So you're going to place each allele next to an open box on the top or side, then cross inside of the square. When you have a big and a little letter, try to always put the big letter in the front. But other than that, how you place them in, I could have placed this one first like I did, or I could have placed this one first. That doesn't matter. As long as both of the right letters make it in there, doesn't matter. When you're done, you want to write out your genotypes. So go back, look at all the possible different outcomes you have in there. Write those down. You can see big B, big B, big B, little b, and little b, little b. And then what I do when I do mine is I just zoom in so you can see better. Nope. There we go. Anyways, I express mine as fractions just because when we move over to dihybrids, it'll be easier for you. And so I have 1 of the big B, 2 of the big B, little b's, and 1 of the little b. And then you go down phenotype, write out the physical descriptions. There were 1, 2, 3 out of 4 with large beaks, and then only 1 with a small beak. Zoom out, scroll up. So then I kind of wrote the same thing out using kind of a different problem here, using the same ospreys that we used over here. Let's cross big B, and big B, little different. We've got 2 big B's here. So you're going to draw your Punnett square. You're going to move the alleles to the top next to the boxes and to the sides. Cross inside of the box. Take big B, bring it down. This B, bring it over. This B down, this B over. Bring this B down, bring this B over. So on, so forth. Once again, this up here, the ordering of these letters doesn't matter. Just make sure that your big letter is in front of your little letter down here. Once you got that figured out, come over, write out you do your genotypes, figure out you've got 2 of the big B's there and 2 big B, little b, so 2 out of 4 and 2 out of 4, 50% for both. And then there are no little b's, so you only have a large beak phenotype, which is going to be 4 out of 4 for that. I'm hoping that you have some experience with this and that you've done it before, so it's not super confusing. Trying to get rid of the blue box. There we go. Haha. So when we cross for one trait, it's going to be a monohybrid Punnett square, and that's going to be a 4 box. You're only doing it for one trait, which would be represented by B. Those are pretty simple to figure out. When we do a dihybrid cross, whenever we cross two factors of traits, it's called a dihybrid cross, monohybrid, dihybrid. So you have two sets of letters going on here. The way that you figure out what goes on the top and the sides is you're going to FOIL out to get the positive. This right here says just determine them. But if you use the FOIL method, where you go first, outer, inner, and last, right there, you can figure out the pairing. I've done that here where we've taken the first T and the first G and put them together. And then we took the first T and the last, or the outer two, the ones on the outside, big T and little g. And then you've got the little T and the big G, the inner, and then the little T and the little g, which are the last of the last letters of each pair. Do that, and then you can stick these on the side. It doesn't matter which one goes on the top or which one goes on the side. Just as long as you have a T and a G for each box, like you see here, because each trait needs to be represented in the cross. All right, when we're bringing the letters down and crossing them, don't mix them up, try to keep them together. So do not write TG, TG, write TT, GG. Keep the traits together. It's gonna be more easy on you in the long run. You can see the example here. Once again, if you're going back and you're looking at this while you're doing the practice, should make sense. But once again, if this is confusing, I am hosting a special Zoom practice so that you can watch me do it, ask questions, do it with me at that point, not just watch me on the video. And so if this is confusing, I would encourage you to come to that so you can learn more. But don't mix them together. Keep the traits together. Fill out, cross your box. Don't forget to go back and figure out your genotype and phenotypes. I'm going to give you a little hint. When you are crossing two heterozygous in a dihybrid, so that means that, you know, the alleles are different. So you have a big letter and a little letter for each one. So this only works if both traits are heterozygous, like they are right here. And in this situation right here, so I'm looking at these two crosses. This one appears different, but the F2 generation here and this box here were both heterozygous crosses. If you look here, once you fill it out, as long as you line your letters up on the tops and sides, this way it makes a pattern. And so all of the ones starting from your top right going down to the bottom left, those are all going to be heterozygous, and there's going to be 4 of them. And then all of the ones going this way are only going to appear one time. So there'll be 4 of them that appear one time. And then your ones that appear like here and here, here and here, here and here and here and here, those all appear twice. And so your genotype and phenotypes are always going to be the same when you have a two heterozygous in a dihybrid cross. And so you're always going to have, for the genotypes, 4, 2, 2, 2, 2, and then 1, 1, 1, 1. And then for the phenotypes, it'll always be 9, 3, 3, and 1. Whatever the most dominant is to the most recessive. It doesn't matter if I'm crossing tall plants and short plants, green and yellow. It doesn't matter if I'm crossing blonde hair and blue eyes with brown hair, brown eyes. It doesn't matter. Whatever the traits are, little hint, secret, this is always going to be your outcome. And then there's a pattern. You can go back and study this inside of that. Now, in this situation, if it's ever all recessive and all dominant, they're all going to be heterozygous. And so if you can just look for patterns in these things, try to it'll help you figure them out. It'll make it so much easier. And then it'll also save on time when you're doing them, because you just can fill things in and not have to worry about it. Down here is a pedigree that I just threw in. If we were in class, we would go through and do these and learn how to do them. But just for the sake of time and things, we don't have time for them, and so we're not going to do them. But I just wanted to show you what they were. They're a way to see how genes are passed down through generations, from family member to family member, to see how you got Karen and Dan here, and so how they passed off. Um, let's see, Dan is a carrier, and so now his son is a carrier. This is obviously an unaffected female and an unaffected male. So these two didn't get it, but Dan passed it off to his son Ken, who then passed, because Ken and Barbie both were carriers. Sally had no chance, and so Sally is going to be an affected female in that situation for cystic fibrosis. And so you can look and trace things back through your family using pedigrees and Punnett squares. Well, I was trying to keep this one short. If you have any questions or comments, you can email me, or just try to reach out, ask questions. Make sure you go through, read this, and I hope that you have a great day. Make sure you watch this. Make sure you read through. I'll open the quiz up. I'll try to open the quiz up later this afternoon, but hope you enjoyed it. It's been a great year, and that's all I got for us.